Okay, so in the spirit of reconciliation, Civil Society of Australia acknowledges the traditional custodians of country and their connections to land, sea, community. We pay respects to elders past and present and emerging, and we, sorry, extend our respect to all those present at the meeting today. Okay, so thank you all for coming to, I guess, the first SSA Canberra branch meeting for 2024. My name is Francis. I'm the current Vice President for the Canberra branch. Just a bit of housekeeping. For those who are joining online, if you can keep your audio muted, that'll be great. We're going to leave all the questions until the end. And if you're online, you can ask your question by raising your hand or asking in the chat. For those obviously in the studio audience, you can raise your hand, and we'll endeavor to try to repeat the question so that everybody, both present in person as well as online, can basically hear it. Just quickly about a bit of a spiel about SSA. SSA, or South Society of Australia, is kind of the national body that represents statisticians, data science from all across the country. And if you want to know a bit more about the various stuff, workshops, events, all that cool stuff that takes place, including the workshop tomorrow, then please visit www.statsoc.org.au to find out more. So today it's a great pleasure to have Professor Nick Golding give us the Canberra branch lecture. Nick is a statistical ecologist and infectious disease modeler with a focus on globally important pathogens. He leads the infectious disease ecology and modeling group at Telethon Kids Institute in Perth. Although you live in Fremantle, right? Fremantle, yep.
Important distinction.
Where his research work combines mathematical and statistical modeling, ecology, public health, and research software engineering. Today he's going to be talking about Bayesian semi-mechanistic modeling with Greta for ecology and epidemiology. Take it away, Nick.
Thank you very much, Francis. And I'd like to extend Francis' acknowledgement to the Whadjuk Noongar people, where my work in Western Australia is based, and also to the Wurundjeri Woi-wurrung Bunurong people. The University of Melbourne, we also have an affiliation. I do have a slight bit of feedback behind me, which is very off-putting, but hopefully none of the rest of you can hear it. I'll try and deal with it, like Francis did. So yeah, thank you very much for having me. I'm very happy to be here. And I'll first of all give you a little bit of a background on me. As Francis said, I kind of call myself a statistical ecologist sometimes. My undergrad is actually in ecology rather than statistics. I did an RA position just doing field work and lab work on biting flies and mosquitoes. Then my PhD was supposed to be just doing mosquito ecology, going out into the field, collecting things and doing a little bit of statistical analysis. And then I kind of fell down a statistical wormhole. And the PhD was essentially in statistical ecology, particularly applying lots of Bayesian methods for mapping the distributions of species of mosquito. So then I did some postdocs doing disease mapping and applying SDMs, are species distribution models, so essentially environmental correlative models to predict the distributions of things, and model-based Bayesian geostatistics. And then I moved over to Melbourne in 2016. All that previous work was in the UK. And then worked at the Quaco Research Group, which does some statistical ecology, working on methods, kind of bridging the divide between health applications and conservation applications, basically because I find statistical ecology fascinating, but I really want my impacts to be in the health science space. And then I figured I could finally tie those together in 2020 by moving over to Perth to work in the Malaria Atlas project, which had recently moved to Perth as well, and then got completely distracted by COVID. So I'm going to talk about a bit of a mishmash of some applications, but the common theme is the type of modeling approach that I take and my group takes, trying to unite mechanistic understanding of processes, so transmission of infectious diseases or the processes that lead a species to be in a particular place, unite that with statistical modeling for the very many parameters that we're very uncertain of and often need to model as flexible spatial or temporal terms. So the talk contents for today, I'm going to just talk a little about my team, the infectious disease ecology and modeling team, what we do, kind of the scope of the problems that we apply the semi-mechanistic modeling to. I'm going to give one example of some fairly high impact work that we did during the COVID years on semi-mechanistic model of COVID-19 transmission rates. I'm going to give a little bit of a demo of the greta package, which is an R package I developed for general purpose Bayesian modeling, but that really underpins all of the semi-mechanistic models that we've been working on. And it's the reason I'm in Canberra teaching a course tomorrow. So some people in this room will be at that course. So you'll get a little bit of repetition, but hopefully not too much. But since I think that's possibly going to be the more interesting to many of the people in this room, being statisticians, I'll just give a few kind of hints towards some greta extensions that we have. So the greta R package has specific extensions for different types of statistical modeling and some applications of that. So first of all, this is the infectious disease ecology modeling team. So we're based at Telethon Kids Institute. And what we do is mathematical and statistical modeling applied to really using ecology to impact public health, but particularly on global infectious disease research. It's a bit of a mishmash of the ecology there, but that's the problems that are interesting. And there's definitely a bit of a gap in terms of bringing ecological thinking into public health, because there are a lot of diseases that either require the same types of models, analysis approaches, or have a fundamentally ecological aspect to them. So this is the team. One of the things that we've been working on very recently, an example of bringing the ecology and the public health together, was reactive work. We were commissioned by the Commonwealth Department of Health to understand what was going on with Japanese encephalitis virus in Australia. So for anyone who's not familiar, in 2022, suddenly a whole load of detections of this virus were made in Victoria and New South Wales, and then subsequent detection in South Australia and Queensland. And very recently in Western Australia, there have been some detections, not in humans, but in animals. And so Japanese encephalitis virus, as the name might suggest, circulates in and around Japan and some neighboring countries. And it historically has not been present in Australia at all. There are a few detections in birds right in the far north of Queensland. And it suddenly appeared, and it was all the way down to the southeast corner of Australia. So trying to understand where it actually was, because it's a relatively rare disease. It killed upwards of 10 people, but a lot of people who get symptoms would pass it off as flu or something else. So where it actually is, but also where it could get to, because it just arrived. We have no idea whether it's going to stick around or it's going to spread. And so that really involves some spatial modeling work to try and pin down the various aspects of its ecology. So Japanese encephalitis virus is transmitted between mosquitoes and birds in its normal cycle. And then it jumps over from a mosquito biting an infected bird and then biting either a human or a pig. Pigs can get severe infections as well. And so understanding where it could be really requires us to understand where the possible bird species that can be infected are found, where the vector species are found, and then isolating from the intersection of those where the wildlife reservoir is. So that requires some fairly standard spatial statistical modeling using species distribution models. And then the other problem is understanding where it is at present. And that really is dealing with a number of points where it's been detected and a very variable effort in surveillance for the disease. There has been surveillance where people have gone out and screened mosquitoes and also screened livestock. And obviously, we detected in people as well. And the effort from the perspective of where you might see JV detected really depends on the number of people and the location. So that leads us essentially to a thinned inhomogeneous Poisson point process model for transmission intensity. And then combining those two things, we have an idea of both where the pathogen is and where it could be. And unfortunately, I can't show you any nice maps of the result of this because just over the road there, the Commonwealth Department of Health, they're very hesitant to share any of the data or the results from this, just because it's a little bit politically sensitive and involves some commercial interests in the piggery work. But that's an example of the type of work we do, this sort of spatial modeling. And when we're not doing that work in Australia to support Australian health, we're also doing a lot of work on supporting malaria control in Africa. So the Malaria Atlas project is a big spatial modeling, mapping, data collection project and exercise that's based in Perth. And we have another project called the Vector Atlas, which has a similar aim of collecting lots of data on where malaria mosquito species have been seen in Africa, all the data that's available to determine their distribution, and then trying to make spatial maps and products that are useful to people who are involved in control of malaria. So normally the national malaria control programs there. And so unfortunately, the state of data on where malaria mosquitoes are is absolutely terrible, despite a huge amount of research investment. The collection of data hasn't really been prioritized, particularly not the entomological work. So there's a lot of similar sorts of efforts to understand the environmental conditions suitable and where effort has been put into detecting things. So similar ecological spatial distribution modeling, but particularly including more mathematical modeling of the life cycle of mosquitoes to try and bolster the data, with some other ways of knowing where the species could potentially be. And a huge part of our work for the last 3 years has been using statistical modelling to inform Australia's COVID-19 decision-making. So as of March 2020, when COVID started arriving in Australia, our team were commissioned by the Australian Department of Health to support decision-making. And so our team was essentially in charge of estimating rates of transmission in all the different states and territories in Australia over time. Also estimating a number of other parameters, such as the transmission advantage of new variants as they arrive. And that information was fed directly to the state health departments via the Australian Health Protection Principal Committee, and also occasionally to National Cabinet directly, where there were major decisions to be made. There's a former Prime Minister pointing at one of our graphs there in one of the kind of press conferences that was obviously more prevalent right at the beginning of the pandemic. But that also, those estimates fed into a number of scenario modeling and other analyses done by other mathematical modeling groups, and also into some forecasting that we did a lot of work on. Up until December of last year, we were still contracted to make forecasts of the numbers of cases and hospital admissions that would be expected in each state and territory. So that's taken a huge amount of our time. COVID has been over for most people for quite a while, but officially in Australia, it only finished last year when the emergency declaration finished. But obviously this capacity to have modelers, statisticians doing this work is something that doesn't exist as a core capacity in government. And during the pandemic, the approach was just to lean on the academics who've been doing this work a bit in the past. And then a whole group of academics, including us, had to essentially divert research staff away from their normal research projects onto these consulting projects, and disrupt their research careers in the process. We're trying to put that on more of a level pegging now by setting up a consortium so that we have a clear organization of academics who do epidemic forecasting and analytics. And so this is a brand new thing that we're setting up. We're currently in the process of persuading the federal government to fund it, either through research grants or some other means. But going forward, hopefully this will have a kind of a stable home for making these kind of analytic supports for government. And another really key component, Dr. Nick Tierney here is just in the room over here, he's waving. So Nick's a research software engineer in my group, and Dr. Saras Windecker also joined the group recently, and mostly in a research software engineering role as well. A lot of what we do relies on software for statistical modeling. And so Greta will be talking about, that's essentially the core Bayesian modeling software that underpins most of the work that I've just talked about there, and we'll talk some more about. But we also do all sorts of other little bits of interesting bits of software for statistical analysis. And so one of them was quite informative or influential during the COVID modeling work, was actually to support mathematical modelers who needed to understand contact patterns between different groups of people in Australia and overseas, and also kind of split out by different locations, workplaces versus at home, and different sectors of society, more economically well-off and less economically well-off. So contact matrices are the classic way that that's described. So this matrix here has different age groups in bins on the rows and the columns, and then each cell is the kind of the rate of contacts between two individuals of those two different age groups. And they have these interesting patterns. People tend to hang out with their own ages, which is why there's a diagonal stripe. And the off-diagonal bits there are about one generation apart. People hang out with their parents and children. So basically, there was a need that we identified. Those things need to go into mathematical models, but people didn't have a good way of estimating them. So we've had a kind of a side project that Nick's been leading on statistical estimation of these things, so that they can be used for more different applications, and turning that into software. So that's a very broad overview. I just wanted to give a more specific example of why this semi-mechanistic modeling has been particularly useful in Australia, particularly for COVID-19. So we learned a lot during the pandemic. Before the pandemic, the state of the art in estimating transmission rates was a set of models called discrete renewal models, which are relatively straightforward. And the aim really is to estimate the effective reproduction number, which is on average over all the cases that we have at the moment. How many new cases is each case causing? If that number is above 1, the epidemic is going up. If it's below 1, it's going down. And there are a number of useful things for that, including calibrating epidemic models. And the standard way of estimating this is with a statistical model where you essentially take the number of cases at one time point, and then you take the number of cases at a future time point, and then you divide those two. Of course, the future time point there is when these original people would have infected the others. So you're dividing the number of new cases by infectees by the infectees by the infectors. Obviously, we don't know the single gap. There's not always everybody infects people 3 days later, for example. There's a distribution. So actually, discrete renewal models essentially use a convolution of infections over time as the denominator, and then the new infections as the numerator, and there's a statistical model that goes in between to estimate the rate, essentially. And the standard prior to the pandemic was to use a Bayesian model, but a conjugate Poisson gamma model, where you have a prior for effective reproduction number, and then you have this Poisson rate, and then you show it this data, essentially the rate of infectiousness and the number of new infections. But this is just essentially a one-off deterministic calculation at each time point. And there was software essentially for estimating that within periods and then doing some post-hoc smoothing. And so this is the output that was presented to the government right at the start of the pandemic there. And this was just after the lockdown. So what we're seeing is in Victoria, on the 1st of March 2020, the estimate in Australia of the effective reproduction number was just below 2 on average, but with a bit of spread, obviously. And then those vertical lines that you should be able to see are lockdown measures. So the first and the second major intervention that came in there. And as we saw, the number of cases came down very rapidly. Of course, the plan was just to suppress cases, but what actually happened was we accidentally eliminated COVID in Australia. That actually poses a problem for statisticians. It's great for public health people, but if you're asked to estimate what the effective reproduction number is when there is 0 cases detected or close to 0 cases detected, you have a big problem because you're just relying on the data itself. And so this model, because it has a prior of the effective reproduction number of 3, which was estimated from international data, in the absence of data, the estimate trends up, which is what you can see happening in that red box there. And that was a point of confusion for our policymakers. And it's completely sensible if you understand the model, and very difficult to communicate why the thing is doing this otherwise. And of course, if we didn't have a prior, if we had some frequentist method, we would have just a big explosion to the point that we have a degenerate estimate and we can't actually provide an estimate. So the problem remains, what should the estimate of the effective reproduction number be? And one answer is you don't need to worry about it because you don't have any cases, but people were very worried about it. They were worried about what would happen if we were to allow more people in. We knew people were coming in all the time. And what would happen if we were to relax restrictions. And so the question actually was, what would the effective reproduction number be if we had large-scale transmission? And how can we essentially tell the model that we think it should be low because we have these interventions in place? It should carry on from this previous place. There are some statistical approaches you can take there, but what we did was take a slightly more mechanistic approach to inform what we think the R effective should be when we don't actually have the case data. And so the mechanism behind R effective, you can break it down quite simply into 2 parameters. So the effective reproduction number is the number of new infections per current infection in essentially a train of transmission. One way of modeling that is to say that each infected person has a number of contacts, and then some proportion of those contacts are infected. And the utility of this is that you can break it down into something we can measure. We can measure the number of contacts that people have, at least with surveys. We can ask the general population how many contacts they were having at that time. And we can also, if not perfectly estimate the transmission probability, we can back calculate it, and we can say we expect certain measures, some of the new interventions that were being brought in, telling people to socially distance and so forth, would reduce the probability of transmission. So we have these 2 things. We can start to build up a little bit more mechanism around that. And so we essentially developed this relatively within an infectious disease modeling space, simple model of the effective reproduction number by splitting our transmission firstly into household and non-household situations, and in each of those multiplying the number of contacts by a transmission probability. So that's everything that's going on in the parentheses there. So don't know if you can see my cursor. Yep. We then essentially have a parameter here for the number of household contacts, and I would say don't even bother try to read the notation at the bottom without the definitions. We have the number of household contacts, which essentially doesn't change over time. That's pretty constant, and we can measure that from the ABS and information on household structure. There is an estimate of the transmission probability in their household, which largely depends on how much time people are spending at home. Other than that, people weren't following particular measures in their household to prevent transmission. We also have a parameter here for the number of contacts that people were having outside of the household. That does change. Obviously, when we brought in the lockdowns, everyone was staying home and avoiding people. The number of non-household contacts was the main thing that was reduced by doing that. And we were able to inform that with a separate source of data. So we had been running some behavioral surveys right just before, essentially, just before those lockdowns came into place. And we estimated the number of contacts people had then. And then we managed to persuade the Department of Health to fund repeat weekly contact surveys, just using a polling company to ask random members of the public how many non-household contacts they had. They were asking a number of other questions as well, including what sort of preventative measures they were taking, what proportion of them were avoiding contact with other people. So we then had two separate data streams with which we could inform these two major aspects about.
Data store thì nguội hơn là chip thì nóng hơn.
So we have the number of non-household contacts here and the non-household transmission probability. We can track both of those over time. Obviously, the probability itself needs a bit of extra modeling. Then there's one extra effect that was really important in Australia, which is the effect of finding cases quickly and putting them into isolation. It turns out, I think that was underestimated by most people, how important and impactful that was. We really managed to knock down transmission quite a lot by a really intensive contact tracing and jumping on transmission. So we actually can measure that as well. We have data from the cases on how quickly people are out in the community. So this is a big diagram, but essentially what we have is a number of different data streams. So we have data on mobility. So Google providing really nice data, and that was informing our modeling of the number of non-household contacts people had. We had data on people's behavior, and we had information on how long it took to detect cases. So those are what those time series look like. These are estimates of non-household contacts and proportion of people adhering to hygiene. Jumped right up with the first lockdown and then started to wane as people relaxed a little bit more. And it's waned quite a lot recently, as you'd imagine. And then these are distributions, essentially, each point in time of the time it takes for people to be detected. So each of those then feeds into a different component here, and for kind of the population average of those parameters. And all of those feed into that previous equation I gave you on the expected effective reproduction number. So we make a clear distinction between that expected value under this mechanistic part of the model, which is based on population-wide parameters, the whole population averaging over them, the number of contacts that it had. We make a distinction between that and then the observed, which we know is going to differ from that for a few reasons. A, because stochasticity, small numbers of samples, but B, because actually the population that are infected will behave slightly differently to this entire population, and they might come from different sections of society. But this is the mechanistic component. And then we combine that with a statistical term. So this is the mechanistic term, and this is a statistical term, which is a time-varying random effect. We use a Gaussian process for that, and that essentially is a time-varying residual. And then we say that the effective reproduction number that we would expect to observe is a combination of these two things. And I won't go over exactly how we combine those, but it's essentially just a residual term. And then using the same likelihood as before, a discrete renewal model where we account for the generation interval distribution and then therefore can contrast the numbers of infections, of new infections on a given day with the number of people who would be infectious, then we can inform this model. And it actually gives us our estimate here of the effective reproduction number. This is for New South Wales, this trend here. So this was the number of locally acquired cases right at the beginning of the pandemic, coming down to very little, next to nothing, just a few blips here and there, and then some of the smaller outbreaks that happened later. So we have our estimate here of the effective reproduction number. Where there's data, it's really closely informed and has very precise estimates. And in this period here, it falls back on the mechanistic part of the model to infer what the effective reproduction number would be if there was much transmission. And then it smoothly goes back to estimating from the data here. So this is essentially the observed effective reproduction number. This is our estimate of what transmission rates were or would be. And this is our estimate of kind of the population-wide average using that mechanistic model over. And then this pink thing here is the statistical Gaussian process bit. That's the difference between the two. And there were times that the chief health officers and chief medical officers were interpreting this as well, because the presence of a blip here actually has some meaning in terms of understanding that the population that's doing the transmitting is probably different from the overall population. They're either higher transmitters or less transmitters than the population average. So this work was published. All of this was done with a computational pipeline, bringing those initial kind of data streams, and then a Bayesian statistical model that estimated the mechanistic part and also the Gaussian process part for the semi-mechanistic. And obviously the impacts of that were via the health departments, as I mentioned earlier. And so those trends, those estimates over time, particularly of what the effective reproduction number was likely to be in the event of an outbreak, were used to determine whether to implement lockdowns or open lockdowns in a number of states. But those estimates as well from the mechanistic part of the model, calibrated to that data, enabled us to make predictions as well of what the transmission rates would be under a number of different scenarios, which was incredibly useful when we were tasked by the government, the Commonwealth government, to help plan the vaccination scenarios for the national plan. So this was the Doherty report modeling, which got bashed around as a political football, which was fun. But essentially the estimates of what the transmission rates would be like under those different scenarios, they come from this diagram here essentially is an upside down bar chart of the contribution of different components to reducing transmission. So this is the rate of transmission without any intervention. And then this is the one line. If you get it below 1, you have control. This is all essentially what people were doing just from the behavior as it was and from limited contact tracing. This is the effect of vaccination. The more people you vaccinate, the bigger that effect is. And that obviously uses Nick's work on the contact matrices to estimate the impact of vaccination, accounting for population mixing. And then these are essentially different levels of severity of lockdown that would be needed to bring things under control. And so this work essentially informed the government the Morrison government particularly wanted to open at 50% before they commissioned the modeling, because the US and the UK had done so. The US and the UK had a lot more population immunity. And so this work was quite influential in persuading the government to wait until a higher rate of vaccination before starting to relax things. Okay, I'm going to have a sip of water and then pivot to talking about Greta. So the work that was there obviously involved a whole load of data analysis, standard things that statisticians do dealing with knotty data ingress and tidying data and avoiding pitfalls of biases in data, and fitting a range of different models as well. We used a number of generalized additive models and so on, but to fit these semi-mechanistic models to bridge that divide between the mechanism and the purely statistical, we really need software to do that. There is a bunch of general purpose Bayesian software for fitting these types of models where you don't need to worry too much about the inference method, because they will let you code up the model quickly and then run an efficient MCMC engine. So Stan's available. You can also use JAGs and bugs, and there are a number of other things as well. We use Greta, which is the package that I developed starting in about 2016, 2017, which uses Google TensorFlow in the backend to do computation, enables you to do Hamiltonian Monte Carlo. Because it uses TensorFlow, it's actually quite scalable. It has just a different computational model to things like Stan, enables you to essentially efficiently parallelize to very large numbers of chains, but also parallelize large operations like big matrix multiplication or matrix inversion, which, because I really like a Gaussian process, comes in very frequently. And so we have the Greta package to do that. And the aim is that you can write your model in R and it will go away and do all of that and abstract away the computation. So I'm going to try and switch screens here and do a little bit of live coding just to show you how Greta works in the interface. And so what I've got here is, sorry, we've got just a simple linear regression example here, 100 observations, 100 values of a covariate value, and then just simulating some Y data from a linear model. The way Greta works is that instead of defining it in a separate language like in Stan or JAGs or bugs, where you go away and you write a separate language file, and then that's parsed as a separate thing. We actually do it interactively in R. And so it works by building up the generative Bayesian model. So you define the priors, first of all, over your variables, your parameters in your model. Then you use those to construct an object that represents the linear predictor in this case for the linear model. And then you define a distribution over data. So I run this line here. It has to do some connection here with Python to connect to TensorFlow, which only takes a second. Should only take a second. A bit slow today. And then we have this object here, int. So we just defined this as a variable which has a normal distribution with mean 0, standard deviation 5. And that's a scalar. And it's a scalar with an unknown value. But we can move it around. It's called a greta array because it's just like an array, just like in this case, a 1-by-1 array. So a scalar. And you can use it just as if it was a native R numeric object. So we create one for coefficient, one for the standard deviation as well. They all look like that. And then X here is just an R vector, just 100 values. So just as if we were multiplying a scalar by a vector in R, if we multiply those two things together, the result is an object, a variable, which represents what you would get when you take a normally distributed variable and you multiply it by these values in the vector. So we get this unknown object, which again, we can do things with. So to find all of these and to find the mean there. So that's just an object we can do things to. And then when we want to complete the model, we essentially say that y is a variable that we observe. And we say that the distribution over these observed values, remember that's just a vector, is normal with the linear predictor and the standard deviation. So it's just the definition of a linear model. So those objects there, the mean and the intercept and the coefficient, those are all kind of connected to one another by what we just ran there, how we created the different objects. We can pull them all together with the model function, which gives us this model object, which isn't very interesting on its own, but it's an object that we can do things to. We can do numerical optimization if we want to find the parameters of these values that maximize the posterior in this case, or we can do MCMC, which is normally what we end up doing. And so this is a very simple example. It's linear regression. Obviously, the power of MCMC and having a nice language which lets you just focus on the model itself is to do much more interesting things than a linear model. If you want to do this, you can just do a frequentist or a much simpler Bayesian solution. But that gives you then our draws. It says here that
break a new teacher
on 8 CPUs.
Yeah, okay.
So that's kind of just demonstrating the fact that under the hood, it's not doing the normal computation where you split it up and you do essentially one chain on one CPU. It's actually doing them all simultaneously and parallelizing it, which becomes really powerful if you want to do ridiculous things where you have really, really large arrays, or in this case, if you want to run a really ridiculously large number of chains, it's going to run them all simultaneously and then parallelize that operation. And that's because TensorFlow has this tensor model where you're essentially, for every one of these operations, you're adding an extra dimension. So in this case here, where the coefficient is just a scalar, it turns that into a vector of length 400, and it does the same across all of the other objects in the model and solves the whole thing in parallel simultaneously across all of these things. I'm actually not going to, I'm going to regret doing that, because if I try and run a ggplot with 4,000 lines, it's going to take absolutely forever. So I'll just run that again, and you can get your samples out. And so then the outputs are in a coda format, and just like many other MCMC languages, so you can do your normal Bayesian things with them. One of the other really nice things about Greta is that once you've got this model defined, this is essentially the model defined in a kind of upside down version of how we would normally write out the statistical notation for it. You can do other things as well. So because it's interactive, I can say I want to calculate what the value of the mean, maybe just like the first 3 elements of the mean would be if I fixed some of the values. So if I fixed the intercept is -2.
Mở tủ lạnh ra lấy ice cream ăn.
And then the coefficient to 1. Sorry, it's a little bit narrow there. It'll go away and it'll calculate that for me. If I wanted to just set the intercept to -2, for example, but then sample everything else from their priors, I can do that. Just ask for 100 samples of it. So that's prior sampling. The other thing I can do is instead of using that, I can just pass in, or what I can do is I can make a new prediction to a new set of data. So X new is going to be from, what did I do it? Random normals. Make a plot of this. So if I want to predict the new value of the mean, I can just calculate it after the fact using these old greta arrays that I've already estimated some values for. I can do that prediction and I can plug in the posterior samples that I have in drawers here. So then I've got 100 posterior predictions of that. So you can actually split apart the model fitting and the prediction into two separate steps, whereas in a number of other languages, you'd either have to implement that yourself as a whole other load of code in that language, or you'd have to put in the data to predict to at the same time you're fitting the model, which tends to break a whole load of analysis workflows. There are many other things you can do with calculating, including posterior and prior simulation. So I think it's quite a nice, neat, tidy way of specifying models. And there's also, that's my emails, you don't want to read that. There's also a website here with a bunch of different examples of different types of models you can do to help you get up to speed, including a lot of the things that are in the original bugs manual and some things that the Stan folks have defined. And then there's also a number of extension packages as well, which is what I'm going to talk about next. So as I mentioned, I'm a big fan of Gaussian processes. We use Gaussian processes and all of our semi-mechanistic modeling and in Bayesian geostatistical modeling. And that could be a bit of a pain to specify. So in Stan, for example, if you want to do a Gaussian process, you generally need to define a function in advance, which produces your covariance matrix. Then you need to specify a number of different things to get the function out. But the nice thing about Greta being written in R is that we can just create new extension packages that do a specific type of modeling, provide utility, and that can be a separate thing, managed by a separate person. You can write a nice abstraction of how you define that model. So in the same way that R packages just do nice things like abstracting away all the complexity of a particular problem, like there's the survival package lets you do survival analyses really nicely, even if you could implement them yourselves. I think that's the real power of Greta, is that we have a package management system already, and so we can abstract these things. So what's happening here is trying to fit some wiggly lines through some dots. All we need to do is to define a kernel or a covariance function. This is using the kind of machine learning terminology for how to define Gaussian processes. So they're kernel functions rather than covariance functions. And you can actually use the machine learning notation of adding together different types of covariance functions to construct a different covariance structure. So you define the kernel there. You can add, whoops, went too far. You can add, obviously, Greta arrays for the hyperparameters, so you can do estimation at the same time of all the GP parameters at the same time as the hyperparameters. To specify a Gaussian process, all you need to do is say what you want to define it on, what the space is, and then you pass in the kernel, and that gives you back this f, which is the function. Then you can just plug it into a likelihood, whatever model you have, however weird your likelihood is. And there's also functionality to extrapolate the Gaussian process to new places. So that's something that I use a lot because I don't want to have to rewrite this stuff again and again. One really important application for Gaussian processes in our modeling is spatial random effects, particularly for the malaria modeling. That's a really key way of mapping things like insecticide resistance in our vector work, or the prevalence or incidence of malaria. And so there's really nice software in Inla and some other packages that can do this by using one really nice approximation, this SPD representation of a Gaussian process in 2 dimensions with a particular type of covariance function and return kernel. Now that's fantastic in Inla, because Inla can use sparse matrices and this mesh structure to speed up the computation massively. Unfortunately, one of the issues has been with translating that to doing things like Hamiltonian Monte Carlo is that you can't really do Hamiltonian Monte Carlo really well on those big sparse matrices. You really need the derivatives to be able to do Hamiltonian Monte Carlo, and the derivatives of those sparse operations are generally not sparse, and it's a little bit of a pain. Fortunately, we've cracked this recently. There's a nice paper by Durand et al. Who show that you can take the sparse precision matrix that you would in Inla, and you can reorder it into a low bandwidth matrix. And then there are a number of really efficient operations you can do when doing TensorFlow and Hamiltonian Monte Carlo. And so there's this is a prototype at the moment. We're going to release it possibly in a separate package to Greta GP, but you just use Inla to set up the mesh and SPD approximation. And then you just have a few steps here to define a Gaussian process there. And it has all the same functionality as Greta GP, but that really enables us to crack this problem of spatial Gaussian processes in a computationally efficient manner. And one other really nice thing, again, more for utility rather than a new computational advance, is a really nice interface to how the MGCV package lets you define smooths. So the MGCV package fits GAMs of various different types, but also has this nice formula interface where you can just say, I want a smooth on this, and then I want to add a smooth on this other thing. And it has a whole library of different types of splines. We can just take that bit of defining a generalized additive model over, or rather the spline component of a generalized additive model over some parameter. And then we can plug that into a Greta model as well. So the Greta GAM package provides this smooths function, which lets you define smooths over a certain data set. And so this Z is the response variable. And so obviously this is just the spline bit. So then we need to push it through a link function. And in this case, for a Poisson regression model. So this here is a cyclic cubic spline. So it actually means that the spline joins up again at the start and the end of this 12-month cycle, which is really handy for modeling seasonal data. And it's just a really quick way of specifying those things. There's obviously all sorts of other types of smooths you can do. And so once you've got that, you can also do the same thing as in Greta GP, that you can extrapolate those predictions to new things later. So that's a really nice little addition as well. And then just finally, wrapping up quickly, one of the other extensions or types of model that I'm really keen to fit, that's another example of a semi-mechanistic model and that we can now fit in Greta, is to bring in more mechanism, more demographic population dynamics into spatial modeling. So I mentioned we use environmental niche modeling or species distribution modeling to map the distributions of vectors, and in the Japanese encephalitis virus example, bird species that are involved in transmission. And those essentially work something like this. You have some covariates which are spatial, they're gridded. And then at a set of locations where you have data, you propose some set of parameters, you calculate this PI, which is essentially the probability of the species being present, you make a predicted distribution. And then obviously you do inference on that by defining an observational model comparing it to the data. So standard statistical inference, but the model is just that you have some statistical model for the probability of presence. The type of model that I'm interested in is just splitting apart that probability of presence into something a bit more mechanistic. So this is a slightly crude assumption about how the probability of presence of a species relates to these two vital rate parameters, which are the survival of a species and the fecundity of that species. And then we model each of those two components as being spatially varying according to some covariates. Now, this doesn't necessarily help you at all with making this prediction map. You just want to predict the probability of presence, it's overkill, but it has some utility if you have some extra information about those vital rates, or if you want to make predictions about vital rates to go into some other model, some simulation model, for example. But the point I'm trying to make here is that rather than just having this really simple logit linear model, you could put in something completely wacky and then fit it in Greta. And in this case, this is a matrix population model that we can iterate over time to solve this fun little model. And the code for it in Greta is pretty straightforward. So this is just using a matrix multiply on a design matrix to do the log fecundity. So slightly different coding than before, but that's just a linear model, exponentiate the fecundity, loaded transform the survival for adults and juveniles. And then we construct this matrix. For every single pixel in the map, we construct a matrix population model. We solve it, translate that to a probability of presence, and then we fit that. And in a Bayesian model for all of the continental US, that takes about 90 seconds to do MCMC on that. So it's actually quite computationally efficient for what's otherwise quite a gnarly model. So that's the type of thing that we're interested in, particularly at the moment. And we're doing some work on that for mosquito populations in Africa. And that's a lot of different things there. But I'm at about 45 minutes, so I'm going to stop there. And I'd love to chat and answer any questions. Thank you.
Thank you very much, Nick. I guess we'll start with the questions in the audience, the live in-person audience first, before we move on to any online questions. You all seem to be blown away. Are there any questions in the... Oh, sorry. Yes.
Thanks for a great talk. So model fitting, you've got basically TensorFlow as your backend gen. Is it possible to have that delineator and say replace that with Stan, say? Like, is it really coupled very strongly with that kind?
Well, that's kind of a question for Nick. That's like an interface, because Nick's now the maintainer of Greta. Yeah. We've definitely thought about having different backends. We wouldn't use Stan because Stan's not designed to have that low-level interface. Because what we use TensorFlow for is not building up the statistical model per se. It's just doing the computation that takes you from a proposed set of parameters to the joint density of the model. And then doing Hamiltonian Monte Carlo is a kind of a separate algorithmic step. So we don't need the full thing in Stan. And generally, Greta's faster than Stan for the types of models we tend to apply Greta for, which are these kind of big spatial type things. But yes, but there are many other types of backends as well. And so TensorFlow is now kind of out of date in the deep learning world. We don't use it. We don't do deep learning, but we use the computation. But yeah, so Nick, did you want to talk about any of the other?
Yeah, so like so basically it would involve like a heavy read. There's plans to use Torch. It would be really good to use Torch because it's a lot simpler to install. Python's a great programming language, but a lot of the work with Python is actually to get it set up, to get everything installed properly. And so, yeah, it's like, in theory, down the roadmap, once we get a few things sorted out here with a bit more of a refactor of Greta, totally possible to swap out other parts. So I think like Torch, and I don't actually know the other sort of progressions in that space yet. Outside of the fact that there's a new programming language called Mojo, which is a superset of Python, which also sounds hard to install. But yeah, so plans, but yeah.
Yeah, and your second question was about speed compared to other things.
Well, I was wondering if there's some models whereby it's or if there's a certain model the Stan can't fit and Greta can fit.
Yeah, absolutely. So I actually did a comparison a while ago between Greta and Stan and JAGs on a model that was specifically chosen to make it easier for JAGs. Because JAGs, certain models is crazy fast because it has nice conjugate Bayesian things. And so that was a probit regression model with a bunch of different parameters. I don't have the plot here, but essentially the outcome was for a relatively small model, JAGs was just blindingly fast because it did this really efficient sampling. Stan was okay, Greta was okay. As you increase the size of the data, JAGs slowed down massively. It couldn't handle memory. Stan went on a bit further. Greta was kind of neck and neck. And then anything larger, like quite large matrices, so a large number of predictors and a large number of observations in just a simple probit regression model, Greta was way, way faster for more than, say, about 1,000 observations. And that's just because it can do those big matrix multiplier, which is the main computational cost in that model, really efficiently by spreading it out over whatever CPUs you have. So in those types of models, where you just have a big matrix multiply, or you're solving a Gaussian process, which involves a matrix inversion, TensorFlow and therefore Greta is generally faster. For models where you need a for loop and you want to do lots of little iteration through things, Greta is really slow. Because it's designed to do these big matrix things, there is an overhead every time you do one of those operations. And so doing like a loop slows Greta down massively. Often you don't need a loop. Like there are very few models where you really need a loop, you actually need iteration. And it's possible to often code up efficient-ish TensorFlow code to do that. So the example I had with iterating those matrices to kind of solve them for the growth rate parameter of a matrix population model, which is the dominant eigenvalue, that can be done relatively efficiently in TensorFlow with some iteration as well, but yeah, requires a little bit more work. So yeah, generally big things, it's more scalable, but also, yeah, it does require you to think about it in a slightly different way than you would in JAGS and STAN.
Yes.
I thank you, Charles, because today, half day, I used the greta. And at the beginning, I was a little bit afraid whether I could install it. You provide the code for Python, it actually works. That's good. Thank you for that. My question is, actually, I use also BRMS methods, but also new user for BRMS. I see that they are very good in terms of BRMS and also spend your one also spend. BRMS, they use things, they call it NURBS method, no neuter, but they're also based on ACMC. So I didn't find any comparison between BRMS and greta. So I also tried to Google it. Maybe you can put something in your.
Yeah, cool. I'll repeat the question for the folks online. So
the question that actually comes from my study and work with Robert. We try to create better animal model. The PMS actually works nicely. We try to do something like better binomial random intercept model, but I have only one data for 1 unit, like a smaller estimation. I tried BRMS, it's stuck. I'm not sure whether any way to do this. Yeah.
Okay, cool. So a few questions. So the first one, just a comparison with BRMS, which is another package where you have like an R interface, but you specify more like a formula. So a little bit more similar to like GLMs or LME4, but it uses STAN underneath the hood. Yeah, it's like LME4, and then but it uses STAN underneath the hood. Yeah, so Greta is more like STAN, Greta to STAN, rather than having that formula interface. The really nice thing about BRIMS is you get this, yeah, you can specify models really, really quickly. So the first question was about how HMC compares to NUTS in terms of speed. So NUTS is a version of HMC which has this extra step. It essentially means you don't need to tune one of the parameters, which is the number of leapfrog steps, because it does it itself. There's a really annoying thing to do with the TensorFlow computation model that means it's actually a little bit of a pain to do NUTS. So NUTS isn't implemented in Greta at the moment. It's something that we can add very soon. We're about to release a new version of Greta that will make it easier to do that. NUTS is in TensorFlow just a little bit slower than HMC, and there is a better algorithm that we will hopefully have there. But generally, they're pretty similar. And so in Greta, you might have to tune one of the parameters, the leapfrog parameter, to make it work. And then it should be at least comparable, if not faster. But normally, it's not really the sampler efficiency that is the main thing. It's the computation time for most of these models. And the other question, there was a question about beta binomial model, which I think the answer depends on the model itself. Yeah. 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 So there's a, yeah, it sounds like that's a model specification type thing that I'd love to chat to you about. Like, it's probably possible to fit that model, but might need some tweaking, and it should definitely be possible in Greta. So yeah, happy to chat about it. Yeah, just absolutely. I'll chat to you afterwards and we can exchange details. Yeah.
Are there any questions in the chat? Or you can either ask it in the chat. Sorry, online. You can either ask it in the chat or you can unmute yourself and go ahead and ask. There is a question from Megan. You can.
Yeah, could you clarify which different implementations? Sorry, we've been talking about a bunch of different things here. I'm not sure what that refers to. Do you mean STAN and Greta? Yeah, so that'll depend on the model entirely. For anything with kind of a relatively large just GLM type model, if you've got more than 1,000 data points, Greta probably would take a couple of minutes, and STAN would probably take a little bit longer. Pretty similar, but then when you get to larger data sets, tens of thousands of data points, Greta probably would take up to 10 minutes and STAN many hours. Basically, it's the scaling that's more of a difference. It's difficult to say exactly what the number of minutes is because obviously that depends on the model itself and the computer and a bunch of things.
Thank you. Any other questions, I guess, either online or in person that anybody would like to ask? Yes, one question in the audience.
Well, I guess I'm sort of thinking about some more question, but your examples are there where you specified predefined distributions like normal or whatever. If you had a distribution that was not standard, if you had a function for the density or something, could you do that easily?
Yeah, that's a really good question. Yeah, and so the question was, if you have a weird distribution that's not already coded up in greta, how easy is it to add it? It's deliberately not super trivial, and that's because we made a deliberate design choice at the beginning of greta not to give the user too much control in the model definition. And if somebody wants to define a weird distribution, they need to package it up nicely. And so there's essentially like an extra step. And that's because in kind of the Stan world, you can do that. You can directly modify the joint density, which means you can get yourself in a lot of trouble if you start doing that. And that creates this awkward system where for a beginner, you start writing out the simple models where you write out the generative model, and then suddenly you want to fit a more complicated model. And the answer is to go to Stack Overflow and copy a whole load of text there and copy it in, and then try and modify it to your example and hope you don't break it so that you're actually defining the wrong model and it's all going a little bit wrong. And so we want people to have the ability to do that. We want people to extend it. And so there are ways that you can define new distributions, but we deliberately make it so that you do that as a separate package or contribute to an existing package. And so we have some so for example, greta GP is a separate package, greta gamma is a separate package, and we have I'm not quite sure which stage it is, Nick, but there's a package to add distributions to.
But yeah, the idea is that it will provide some extra distributions. There's some other things. I think we have like zero inflated Poisson or like other like distributions that weren't part of greta originally, but also it would provide tools so you can sorry, we'll have to repeat that, but yeah, like it'll provide tools so you can create your own distributions, because setting that up in TensorFlow is unfortunately not as simple as writing that in
Yes. Yeah.
Yeah, so Nick was just saying, just in case anyone can't hear online, there's a package greta.distributions, which is in development for people to add those things to, and it simplifies the process of adding distributions.
I guess we'll finish with Max's question online and we'll wrap up from there. So you go for it, Nick.
Yeah, absolutely. So the quick answer to the question is, can greta handle any model written in Stan or JAGS? Is there a way to quickly translate the model across the languages? So to the second bit, quickly translate across the languages, there are a bunch of examples on the greta website, which I still have open here, where there are actually the paired things. So this is Stan, and the Stan example is given by Stan people, and then there's the greta example there. So that's a way that you can quickly go and compare between them. And then similarly for bugs, there's example bugs JAGS code, and there's example Stan code, and then greta code. So you can look at those and see how to define them. And you can see for some of these things that like JAGS loops, greta does not loop. So some of those things are a little bit different. And then the other question, or the start of the question is, can you define anything in Stan and JAGS? Sort of. You can do anything in greta that you can do in Stan, because it uses Hamiltonian, Monte Carlo, and automatic differentiation. There are things that you can do in JAGS that you cannot do in Stan and greta. And those are MCMC sampling on discrete parameters. So if you have an unknown variable in your model that is, for example, binomial distributed, so it can either be 0 or 1, your sampler needs to flip between the 0 and 1. You can't do Hamiltonian Monte Carlo on that because Hamiltonian Monte Carlo needs a derivative, and there's no derivative between 0 and 1. There are other solutions, so you can marginalize those things, and there are tricks in Stan and greta, but there are some specific models where you have to do sampling of discrete things, and it's difficult. At some point, we should be able to get something like that into greta so that you can sample some discrete things if you want to. If enough ecologists shout at me to do that, will probably happen, because ecologists seem to love those models.
I'll certainly be shouting at you for that. Okay, so I think we'll finish up. Let's thank Nick again for a very interesting talk.